Thanks, Arun. I think I can just open the floor for questions after Hans's so inspiring and detailed talk about the past, but I think it still is important to go through what we plan to do to continue delivering the genetic gains in the future. So just build upon what Hans has been contributing. So at present, our summit wheat improvement program in Mexico is targeting close to 60 million hectares. That's around 25% of the global wheat area. I'm getting echo here. Do you get? Okay. Uh, and there are four major mega environments, the irrigated, high rainfall, semi-arid, and the irrigated warmer areas. Although these are mega environment which were around 1990s were defined very well, the situation nowadays is you better throw the mega environments under climate change scenario for the future, as Hans has shown already. And so, and to make the to develop the more climate resilient varieties of germplasm, the key issue here is bringing various traits together in the germplasm, what we call core traits, and that ranges from high yield, and as you could see, the water use efficiency, drought tolerance, heat tolerance, of course, resistance to rusts, which everywhere goes, quality, and I think the more important now is also looking from the nutrition side, and thanks to the Harvest Plus program, which pushed the agenda, and now we feel so happy that we could have varieties which have high zinc and also associated increase for iron. And there are various other diseases which we also focus depending on the environment. And slowly over time, I think you have to bring most of the things together. So what in the last 10 years has happened, and hopefully it continues, is through all the generous support from various donors. Hans listed some. I will list all of them at the end. We have been able to upscale our breeding and testing program. This was very well described, the shuttle breeding, and in the BGRI program to address the stem rust situation, we expanded our Mexican shuttle to Mexico-Kenya shuttle. It was debated between Kenya and Ethiopia, but because of the crop season and cycle matching, we ended up in Kenya, where you can grow 2 crop seasons per year and continue with the genetic gains. The other important part, what program is doing, we keep a lot of parental diversity to ensure continuous genetic gains, not just now, but also in the future. We also have targeted introduction of new diversity and utilization around what kind of crosses we make. You can read all this. I think the important part is managing large populations to identify some outperformers that we do through the selected bulk approach of shuttle breeding I mentioned. And I think important part also is to think of simultaneous selection for more than one trait at a time, phenotyping addressed. Some of the new things you will hear in later presentation would be how the genotyping and genomic selection is being used, and the importance of data. All the data which is being generated is being maintained for now and future utilization. Oh, okay. Important, despite all the progress in many genotyping and predictions, I think is still very important is the phenotyping and Obregon, Hans highlighted, you can do side-by-side phenotyping for drought, and because it doesn't rain much. And in the next field, you are probably getting 8-9 tons yield. So it's a very efficient use, and we utilize this facility. In the first year, we test close to 9 or 10,000 lines, depending on the year, under replicated trial. And out of that, we retain around 1,000 and then test them very extensively for under a range of management, high drought stress, moderate drought, early sown heat stress, late sown heat stress, etc., which helps us picking the more stably performing germplasm. So I think this is very important part, that at one site you can do so many things. And once you do that, once you test so many materials, year after year, what I have seen, that once you do that, you get a small proportion of wheat lines, which in the first year of yield trial starts to look better than your check and Jenny. The check is a very strong check called Borlaug 100, to emphasize the importance. It's one of the genotype which has very stable performance irrespective of environment we use. So it's used in the drought, it's used for heat, and of course for yield potential. So if you compare the yield distribution of the lines tested in the last 4 years, we now see a major shift towards the higher side of performance, and that's mainly because now we have several derivatives of this variety of line Borlaug 100 just coming out. So if you use good parents, you have a good chance of getting good progeny like Jenny Borlaug sitting here. And that's what is happening now. So you'll see in the future some good performing lines coming out of Borlaug used as parent. Hans already showed this slide, and the important part, again, I want to mention, you might think the need for the international trials or nursery may be going down, but that is not the case. We are multiplying more seed, we are sending more sets, there are more requests which we can sometime even handle. Which makes you feel that you are doing good job and people are finding some value in your germplasm. That's why they keep requesting. So to measure genetic gains, we also analyze the data, and we completed using all the cooperators' data which has come back to us. Of course, you can measure genetic gains different way. Every country is looking for the lines which are adapted to their conditions in those trials and nursery. But for us, it's also important to see how well our lines are doing. So last year, we published a paper using your data from 27th to 34th SWIT, the Elite Spring Wheat Yield Trial, which is targeted for irrigated area. In those years, it was grown on many more sites, but we had good data from 426 sites, of which 225 sites were from Mega Environment 1, the high production. And when you see over time the gain over the famous Attila lines, you could see the 1.67% genetic gain. Part could be yellow rust, but when you take the constantly changing local checks, we still could show there is a continuous gain over the local checks, and the area for which it was targeted is much more, 0.72% or 46 kg per year. We also completed a similar analysis, and a paper will come this year in Crop Science for the semi-arid wheat yield trial, and that's 11 years of data. More than 700 sites had grown in these 11 years, of which there were 216 sites in the real drought situation, and another 222 or 232, I cannot read very well from here, in the moderate yielding sites. And when you look at it, there's more gains under the high drought stress site, around 1.82% over the mean of the checks. That's around 38 kg, but also the gain in the medium yielding sites and a very similar gain in higher yielding sites. So I think that's very important to recognize that these lines are not only performing under high drought, but they do perform under high yield situation as well. So they are very responsive. We had published a few years back for the heat in early maturing line, and you see a similar genetic gains. So it looks like thanks to the cooperation with so many partners we have, we could show that gains are being made. So on the rust side, which many people know me more on the rust side than the breeding side, we apply a concept of slow rusting adult plant resistance. And the reason for that is the durability. And you know how fast the pathogen is evolving, but we want to break that cycle, and we are using some of these genes which are now very well known, LR34, 46, 67, which we call multi-pathogen resistance genes. So not only rust, powdery mildew, and in some cases there are a couple of publications showing that LR34 also slows down the buildup of fusarium, and in the leaf, the transformed line showed better resistance to rice blast in rice plants. So there's a lot of things we still don't know about them. And the other interesting part is there are many other QTLs which you can use, and consensus genetics showed some of the important chromosomal regions which may have other pleiotropic APR loci. For me, from the breeding side, what we have learned and we deploy for rust and many other diseases, if you have one of these genes alone, you don't get enough resistance. Often people call varieties are susceptible, but if you can bring together 4 or 5 genes, you get very high level of resistance. So a lot of breeding is based right now on this particular concept of putting these 4 or 5 genes together. So we started with leaf rust a long time back, because when I joined CIMMYT, varieties were changed in Obregon every 3 years. And I'm glad to say that the main basically use of APR slow rusting, which I can say now is mainstreamed. Why I say that? Because more than 70% lines which we distribute internationally, shown here as green bars, have a very high level of resistance in the field situation. And because of that, you hear less about the leaf rust problem these days. Yellow rust is a continuing challenge, and I think Dr. Morgan Homoller and others probably will talk about it. If they have not talked about it, I use their publications to show how fast the rust is evolving and moving. There are so many lineages. He reports new one showing up at different places. But what it has done to us is you start to see that disease is picking up much earlier. Resistance is breaking down also much faster. In case of Mexico, since we detected this new race belonging to PST1, PST2 lineage, in the last 12 years, we have a breakdown of so many genes which were used to be effective. And that's where the adult plant resistance, its use becomes so important in the breeding program. but we still have not mastered it. There are some issues because yellow rust appears much early in the crop season at juvenile stage, and that's where we run into problems in areas where disease now is starting up early. If disease comes somewhat later, adult plant resistance works very well. So we basically looking at the use of some other genes which we have characterized and some other places also have identified. I mentioned some of them here. If you put this moderately effective race-specific genes with adult plant resistance genes, you can you get very high level of resistance. And even though in Mexico, when we select, everything is very clean. When we send to Kenya, where you have different population, different environment, or India, just data came from Ludhiana, you could see that some of the lines which are highly resistant in Mexico, they are not so resistant, but they are not becoming completely susceptible. So that means the part of resistance which we select is still surviving in those to those populations of pathogens which are there. So we need to continue making progress for this disease, which I think is a major challenge. Hans already showed this slide. The phenotyping platform in Kenya helping a lot on making progress to stem rust. And I'm very pleased to show you these pictures that finally we can get, these pictures are from Kenya, lines like this, which are so nice and clean, but resistance is not based on a major gene, and the resistance is based on slow rusting APR gene. So what we are distributing these days, 10 to 15% lines are like this. 40 to 50% lines have adequate resistance, which can survive in most of the other areas of the world. We still have 20, 30% lines which have race-specific genes. And to further broaden the genetic diversity, you use marker-assisted incorporation. There are 23 new genes currently being introduced. And there are some stem rust resistance genes, but also yellow rust and some for insect resistance. So this is additional activity going on to introduce new diversity in the germplasm. I also want to make a comment on septoria because it's important in many high rainfall environments, especially Ethiopia. For example, Tunisia, Morocco, in years you have high rainfall. We can get now semi-dwarf lines which are practically immune, almost immune, compared to the susceptible. So Taluka was the place from where in the 70s selection for in semi-dwarf varieties it started. And by Dr. Borlaug that time, because his Siete Ceros varieties were famous varieties, faced some issues. So he put that program. And synthetic wheats are playing very good role to get these lines. When you go to Ethiopia, you also see some smiling faces because some of the lines selected here look highly resistant in Holeta, which is a hotspot site. Okay, so this new challenge in South Asia, Hans once again covered, but we are also what you call latent diversity in your germplasm. Even though you didn't select for it, you have it, and once you phenotype, you find about it. So we now know this VPM translocation initially shared by the French group, Dr. Duchinov. I was lucky to visit him a long time back. Is becoming very widespread in germplasm, and it also has some resistance to wheat blast. But interestingly, not all lines which have 2NS are highly resistant. But through phenotyping, you could find lines like Barigom, which was happened to be a biofortified lines line variety. It is released, and you could see how in the field it looks against the susceptible check. And that's the beauty of the phenotyping platform and its use in the future. So when I mentioned biofortified, this is another success story which I think we need to move further for the human nutrition aspect. We started just around 10 years back using some of these progenitors, the synthetics, the spelt wheat, the dicoccum-based synthetics. There's a few exceptions which had a high zinc. They were used in the breeding program, and we are very grateful that the national partners in not only Bangladesh, but initial two countries, Pakistan and India, they have released biofortified varieties where spelt wheat and dicoccums have been used. And there are some photos here showing these varieties in the field in those countries. So this is something we can do more and try to mainstream in the wheat gene pool. So close to 1 billion people will get benefited in our target environments if we succeed. Bread making, many semid germplasm people were targeting is not so good for the bread making at one time, but that has changed in the last 10 years also. And as you could see now, we have close to 20% lines which are very good for bread making, and another 60% line for flat bread products, and the proportion of inferior quality line has been going down. In fact, it was very low, but our quality man who runs a very high throughput lab, he increased the bar now from this year's data is to bring it up, so putting more pressure on us, which is good in the long run, and this is the kind of bread. And basically what we did was utilizing and trying to use the better glutenins when we choose the parent, and that's in the 10 years we could shift very significantly. So what has been the outcome? So I just list here some 95 direct semid varieties. There are probably another 80, 90 varieties where semid germplasm has been used as parent and released by the partner programs. Just in the, well, I said 2018 because we got some data now. And these are some 20 partner countries where these varieties have been released. You can look at it if you are interested later on. My two colleagues very soon will talk about these two things. I'm not going to mention except that we are trying to integrate genomic selection and high throughput phenotyping in our program. And we hope if these are integrated well, we can accelerate the genetic gains if we find benefit, which we should. And finally, to get all this work, we are very thankful to the Gates Foundation and DFID, and the reps are here. Thank you very much for your support and through these projects, as well as through your funding to the CGIAR or CRP wheat. There are several listed here, several governments who support us directly through projects, and interestingly, some farmers' organizations like AgroVegetal, GRDC, through various projects, and the Mexican farmers. We are able to continue the activity and able to deliver hopefully better germplasm in the future to continue delivering genetic gains. And thank you very much.